Welcome to our continuing discussion of Mendelian genetics. Mendelian genetics. Now we're going to be in Chapter 5, looking at extensions and modifications of Mendelian genetics, where things are a little different and more complicated than they were in our last chapter. First, we'll discuss multiple alleles. That's more than 2 alleles per population. Then lethal alleles. Then genic interactions, how different genes interact to determine a single phenotypic trait. Then penetrance and expressivity, then genetic maternal effects, how sex influences heredity, and then lastly, pleiotropy, where one gene can produce multiple phenotypic traits. So here we are showing a heterozygous genotype, big R, little r. Big R inherited from one parent, little r chromosome inherited from the other parent. So multiple alleles, again, this is more than 2 alleles per gene in a population. And we'll look at a series of alleles determining the coat color in rabbits. So C plus is the wild type, which codes for brown fur in the rabbit. This is the dominant gene. Anytime you see a plus associated with an allele, it's usually dominant. CCH is chinchilla, which means a lighter brown color. CH is Himalayan, which means the rabbit has dark extremities, but the body is pale. So this is where the gene only works at cooler temperatures, i.e. the extremities of the animal's body. And then regular C is albino, where the animal has no pigment at all and therefore is white fur and pink eyes. And these 4 alleles can combine to form 6 different heterozygotes. So wild-type heterozygotes then would be C plus C, C plus CH, and C plus CH. A chinchilla heterozygote would be CCHC. A chinchilla with black tips would be CCHCH. And a Himalayan heterozygote would be CHC, and albinos can only be homozygous because the albino allele is recessive to all the other alleles. Now, null allele, that's a term given to a non-functional allele. In this case, it's the albino allele because the function of this gene is to make melanin, the brown pigment that all mammals, except, of course, albinos, exhibit. And a hypomorphic allele would be a partially functional allele. So it's making some pigment, but not the normal amount. And that would be the chinchilla allele and the Himalayan allele. Both of those are hypomorphic alleles. And then the dominance relations would be the wild type allele is dominant to all. The chinchilla and Himalayan alleles are codominant to one another, and all 3 are dominant to the albino allele. So in a problem like this, you'd be given several crosses. You do each one independently, combine that information to figure out which alleles are dominant to which. So with regard to the Himalayan allele, Siamese cats have this as well as Himalayan rabbits. And in this, the pigment-producing allele is only expressed at cooler temperatures. So if the rabbit is raised at cooler temperatures, like 20 degrees C or less, you get dark extremities like the tail, the feet, the face, the ears. But if a rabbit is raised at warmer temps, like 30 degrees or above, that's too warm for this allele to function, and the rabbit is solid white. They also say you can shave a patch of fur off the rabbit's back and tie an ice pack to it, and then the fur that grows back will be dark. So you'll have a rabbit with a dark spot on its back. Moving on to lethal alleles. These are alleles that cause death at an early stage of development. Therefore, some of the expected phenotypes in the progeny are missing. Lethal alleles are usually recessive. The presence of lethal alleles will alter the progeny, and we get a 2-to-1 ratio, phenotypic ratio, as opposed to a 3-to-1 phenotypic ratio typical of a normal monohybrid cross with complete dominance. So here is an example of a dominant lethal allele in mice. So we have 2 heterozygous yellow mice. We cross those, and we see that 2/3 of the progeny are yellow like the parents, 1/3 are non-yellow, and then And so we get a 2-to-1 ratio because the big Y mice, that quarter of the offspring are dead, and so they don't count. So big Y mice die, and so we have our 2-to-1 phenotypic ratio, and that's lethal alleles. Moving on to genic interactions. So this is when 2 or more genes interact to determine a single phenotypic trait. We call this polygenic inheritance, and there are many, many examples of it. One specialized example we'll discuss is called epistasis, where one gene blocks expression of another gene. The epistatic gene is the blocking gene, and the hypostatic gene is the gene that gets blocked. And this yields phenotypic ratios that are modifications of the dihybrid cross. Not a typical 9:3:1 ratio, but usually only 2 or 3 phenotypic categories and not 4, such as 9:3:4, 9:7, 13:3, etc. So here we're going to look at genic interactions in determining the color of the fruit of bell peppers. This is an epistasis, but it just shows you that 2 genes are involved here. So if we cross a homozygous dominant red pepper with a homozygous dominant green pepper, the green pepper is homozygous recessive. We get, of course, an F1 generation, all red ones, but they're heterozygotes, big R, big C, little c. We cross those guys, and we see we get 9/16 red, big R, big C. Now those dashes mean that the second R and the second C, it does not matter if they are dominant or recessive, the phenotype is still the same. We get 3/16 brown peppers, or big R for brown, little c, 3/16 yellow peppers, little r, and big C, and then 1/16 double recessives or green peppers at little r, little c. So here the R and the C genes have interacted to determine one phenotypic trait, that is the color of these peppers. And this shows the entire cross from start to finish. In P, parental generation, F1, first filial generation, F2, second filial generation, and it's the F2s that show the characteristic phenotypic ratios. Now let's discuss epistasis. And we will use some familiar creatures, Labrador Retriever dogs, where we have a pigment producing gene, and the dominant allele big B produces more pigment, more melanin, so therefore we have black labs. The little b allele, recessive, produces less pigment, therefore we have brown or chocolate labs resulting from little b homozygotes. And this gene gets blocked in some, and therefore it's the hypostatic gene. Then we have a big pigment deposition gene, where big E, dominant allele, allows for the pigment deposition in the hair, but little e prevents pigment deposition in the hair, so regardless of what the B genotypes are, the dog ends up being pale, and that is a blonde or yellow lab. So this is the epistatic gene. So here's our black lab, big B and big E in the genotypes. Here's a blonde lab, little e, and the B's don't matter. And then a chocolate lab or brown lab, which is little b, and a big E to allow pigment deposition. So if we cross our F1 puppies, so here we're crossing a homozygous black lab, big B, big E with a homozygous yellow lab, little b, little e. All our F1 puppies are big E, little e, and they're all black puppies. If we cross them when they grow up, we get 9/16 of our offspring with a big B and a big E, and they're black labs. 3/16 with little B's and a big E, and those are the brown or chocolate labs. And then 4/16 with 2 little E's, and those are yellow labs. So here the little E's are blocking the deposition of the pigment made by the B gene. Therefore, the B gene has been blocked, and it is the hypostatic gene. The E gene is doing the blocking, therefore it's the epistatic gene. Now, there's dominant epistasis, where a dominant allele does the blocking. And the example for this will be summer squash. So we'll take plants that make white squash versus plants that make green squash, and we'll see that the F1s all have white squash, like one of the parents. And the F2s, 12/16 or 3/4 are white squash, 3/16 are yellow, and 1/16 are green. So the white squash have a big W allele, and the other 3 alleles don't matter because it's going to be white. The yellow squash have 2 little W's and a big Y. The big Y allele makes the yellow pigment. The second Y allele doesn't matter, it's going to be yellow. And then the green squash are homozygous recessives, where the little W's allow pigment deposition and the little Y's don't make any pigment, so the fruit is green just like the rest of the plant. So our dominant allele, big W, is epistatic to gene Y. So W inhibits the conversion of a compound A into a compound B, and therefore no pigment is made. Only homozygous recessive little w plants have pigmented fruit. And the Y allele causes yellow fruit, whereas little y keeps the fruit green, doesn't produce any of the yellow pigment. So what this looks like is in white squash, the dominant W allele prevents the synthesis of enzyme 1, and therefore no pigment is produced and the fruit is white. In plants that are little w, compound A is converted to compound B by enzyme 1. And then in big Y plants, enzyme 2 is made, and that converts compound B into compound C, and you have yellow fruit. But in little y plants, the enzyme 2 is not made, and therefore compound B is not converted to compound C, and the fruit is green just like the rest of the plant. And so that's the molecular mechanism going on here behind dominant epistasis in the summer squash. A third type of epistasis is known as duplicate recessive epistasis. And here we're going to take 2 albino strains of snails with different genotypes. One strain is little a, big B. The other strain is big A, little b. And we'll see that the F1s are all pigmented, regular brown snails. And as you'd expect, the genotype is big A, little a, and big B, little b. But then if we cross these F1s, we see that 9/16 of the offspring are pigmented or brown, wild-type snails, and 7/16, just less than half, are albino snails. So what's going on here is we need a big A and a big B allele to make the pigment, and therefore the snail is brown, and the other 2 alleles, the other A, the other B, don't matter. One strain of albino snails are little a, big B, and the second B doesn't matter. The other strain of albino snails are little a, second A doesn't matter, big B. And then those that are homozygous recessive for both are also albinos. So the 7/16 albinos are shown at the bottom here. So the A allele is needed to make enzyme 1, the B allele, dominant B allele, is needed to make enzyme 2. Both enzymes are necessary for the production of the pigment in the snails, and the lack of neither enzyme produces pigmentless snails or albino snails. So recessive alleles at A and B produce no enzyme, hence the term duplicate recessive epistasis. And so here's what's going on. Here's what is going on here. The dominant A allele produces enzyme 1, which converts compound A to compound B. The dominant B allele produces enzyme 2, which converts compound B to compound C, making the snails a normal brown color. But little a snails don't make enzyme 1, and little b snails don't make enzyme 2, and therefore the snails end up staying white. So summarizing epistasis then. Recessive epistasis, a recessive allele at one gene blocks expression of another. Dominant epistasis, it's dominant allele at the epistatic gene blocking expression of the other gene. And duplicate recessive epistasis, recessive alleles at one or both genes block the expression of the phenotype. And this table from your textbook summarizes epistasis. I don't need to go through that. You'll be able to look at that on your own. Then moving on to penetrance. Sometimes individuals that have a genotype do not express the associated phenotype. And that's where penetrance, the concept of penetrance, comes in. It's a percentage of the individuals showing that phenotype that have the genotype. So in an example, we have 42 people that have the polydactyly allele. That would be an allele for having extra digits, fingers or toes. It's a dominant allele. Yet only 38 of these people actually have the extra digits. So what we use, calculate a percentage, 38 divided by 42 times 100, 90%. So we say that that polydactyly allele has 90% penetrance. So 90% of the time, if you have that allele, you will have the extra digits. 10% of the time, you will not. And here is somebody with 6 toes on each foot, 12 toes. Now, related concept of expressivity is the degree of expression of the phenotype. So there's not numbers associated with this. This is not quantitative, but it's more descriptive. So the cleft palate phenotype can be anything from a hair lip, which is just a gap in your upper lip below your nose, like a rabbit has, versus a clefting or a gap in your entire soft and hard palates in the roof of your mouth, all the way running up to your nose. That's the most extreme expression of that phenotype. And polydactyly also exhibits expressivity. In its least expressive form, the extra digits are merely tags of skin, and its most expressive form, they're fully functional digits with muscle, bone, nails, and everything. So that's expressivity. Now, maternal effects. Of course, your maternal parent is your mother, and the egg is an entire cell with a haploid complement of chromosomes, whereas the sperm only contributes a haploid complement of chromosomes to the egg, and everything outside the cell nucleus comes from the mother only. That is everything in that zygote. So here, proteins or untranslated messenger RNA in the egg cytoplasm, not the organism's own genotype, determines the phenotype. So again, phenotype and genotype aren't really linked here. It's a little strange. We're going to look at this in a hermaphroditic snail called Limnia peregrina, where the big D allele is dominant and causes right hand or dextral coiling, given by S+. The little d allele is recessive and causes left-handed or sinistral coiling, given by the little s. So we're taking a dextral snail, crossing it with a sinistral snail. Both are homozygous, S+S. And as you'd expect, our F1s are heterozygous. However, you expect them to all be dextral, but they are all sinistral in their phenotype. Strange. Now, if we self or cross these F1s, we see that our F2 generation are all dextral regardless of genotype. A quarter are homozygous for dextral, half are heterozygous for dextral, and a quarter are homozygous for sinistral, yet all the snails are dextral. Because the mother of the F2 progeny has genotype S+S, all of the F2 snails are dextral. And here's that cross from start to finish, explaining what's going on there. The molecular mechanism should enlighten us some. Spindle orientation during the first cleavage of egg determines the coiling pattern, so you need to review your meiosis and mitosis for this. The S+ allele produces an active gene product, which causes dextral coiling. The little s allele produces an inactive gene product, which doesn't do anything. So what's going on here? Here is a dextral snail from an SS zygote. So the maternal parent was S+S, therefore she has S+ protein in the cytoplasm of her cells, and that's what's determining the phenotype, not the organism genotype, which is little s, which would indicate that this coiling pattern should be sinistral or left-handed. But that S+ protein is left over from mom, and that's what controls the phenotype, the S+ protein in the egg or zygote cytoplasm, and not the genotype of the individual. And that's why this is called maternal effects. Okay, the sex of the individual can influence heredity as well. So sex-influenced traits are influenced by the sex. These are coded for by autosomal genes and expressed more readily in one sex than the other. Autosomal genes are genes on the numbered chromosomes. So for us, that would be numbers 1 through 22, not genes on the X or Y chromosomes. And in some goats, the presence of a beard is dominant in males or billy goats and recessive in females or nanny goats. And in us humans, pattern baldness is dominant in us men and recessive in women. And in women, it's just expressed as a thinning of the hair. Testosterone is required for a complete expression of this gene. So here's some goats. So we have a beardless billy goat with genotype B+, and we're mating them with a bearded nanny goat. And the allele for beards is big B superscript little b. And then we see in the F1s, both sexes are heterozygous, but the males are bearded and the females are beardless. So again, having the beard is dominant in males, but recessive in females. Now, if we cross these guys, we get an F2 generation where 3/4 of our billy goats are bearded and 1/4, just the homozygous B+, individuals are beardless. And in the nanny goats, 1/4 are bearded with the homozygotes only, and the other homozygotes and the heterozygotes are all beardless. And here is that cross, those previous two slides all together. Now, sex-limited traits are also coded for by autosomal genes, but they're expressed only in one sex, not both. So even though both sexes have the genotype for them, only one sex will exhibit the phenotypic trait. So we say that the trait has 0 penetrance in the sex where it doesn't show up. Cock feathering in chickens is an autosomal recessive trait that's limited to roosters only. And male-limited precocious puberty is found in humans, and it's only expressed in males and not in females. So here's a cock-feathered rooster. And by cock feathering, we mean these long, drooping tail feathers, and his genotype is little h. A hen-feathered hen, then she has any of those genotypes, big H, little h, or little h. She never has the long tail feathers present in the rooster. But not all roosters have them. Here's a rooster that is big H, or heterozygous, and he lacks the long tail feathers found in the cock-feathered rooster. Among precocious puberty in humans, here's a male that's heterozygous for precocious puberty, shown by the big P allele. Little p allele is normal, and mates with a female who's little p, and so half their boys will have precocious puberty, those that have the big P allele, the other half will be normal, and the girls will be heterozygous and homozygous as well, but they will all have normal puberty. And if we do a reciprocal cross, the normal female, even though she's heterozygous, and a normal male who's homozygous recessive, we see that we get pretty much the same results. Half the boys will show precocious puberty, half will be normal, and all the girls will have normal puberty regardless of their genotype, because again, this is a sex-limited trait, and it only shows up in males, limited to males. And here are those two crosses from the previous two slides shown together. Here's a table from your textbook showing you how sex influences heredity. Sex-linked characteristics are genes located on sex chromosomes, and those we'll discuss in chapter 4 next, after we discuss meiosis. Then we just discussed sex influence and sex limited. We also talked about genetic maternal effects. Cytoplasmic inheritance is something we'll talk about later, as is genomic imprinting, something we'll discuss later. I believe our last topic for this chapter is called pleiotropy, where one gene has multiple phenotypic effects. And we'll look at an example in humans with mutation in the beta chain of hemoglobin, and that's on chromosome 11, and it's found mainly in people of African origin. Heterozygotes are selected for in tropical environments because they're naturally resistant to malaria, but homozygous recessives suffer from sickle cell anemia or sickle cell disease. And this means that their hemoglobin molecules stack up and cause their red blood cells to sickle, and therefore they can't fit through a capillary like they're supposed to. And so this clogs things up, causing local failures of blood supply to various important organs around the body. So here's a hemoglobin molecule. It's a tetrameric protein, meaning that it's made up of 4 different polypeptide chains, 2 alpha chains and 2 beta chains. And the alpha and beta genes are on separate chromosomes. We're talking about a mutation in the beta chain gene, which is on chromosome 11. The alpha chain gene is, I believe, on chromosome 15. So it's just a single base pair substitution where a T mutates to a C, and that alters 1 amino acid, where a glutamate mutates to a valine, like you can see in the yellow down at the bottom of the slide. Valine is substituted. So the normal erythrocyte is, of course, a biconcave disc. And in people with the sickle cell phenotype, under stressful conditions, their hemoglobin molecules stack up, deforming the cell, making it shaped like a sickle like this or a banana. And of course, that can't fit through the capillary. The red blood cells go through capillaries single file. I envision it like Lifesavers in a little package, and a thing shaped like this is not going to fit through there without clogging things up. So a lot of phenotypic effects from this single gene. There can be damage to our important organs, which have many capillaries in them, like our lungs, brain, kidneys, digestive system. Anemia, the lack of blood cells, can cause heart problems, weakness, fatigue. Our cells aren't getting enough oxygen. There can be bone deformities due to the body trying to lay down more bone marrow to make more blood cells because of the anemia. And again, all these effects are from a single gene. This slide kind of summarizes all the effects of from this disease due to a single mutation in a single gene. So why is this around? Let's look at why it's selected for. Well, the HBA is the dominant normal allele. The HBS is the recessive mutant or sickling allele. Heterozygotes enjoy malaria resistance. So mutation was an A to a T in the beta chain, adenine to a thymine in the DNA, and that caused valine to be substituted for glutamic acid. And the top map shows the frequency of this allele. It's 10% or less in the areas in yellow, and in the areas of red, it's 10 to 20%. So notice how it's mostly present in sub-Saharan and Western Africa, as well as Madagascar, and then a few areas in Southern Asia and in Greece. And then the map below it shows where you find malaria in the old world, mostly in wet regions because it's transmitted by mosquitoes, which need water to breed. And so we can see that there would be selection for it in Southern Asia as well. I don't know if these areas in India are due to gene flow from Africa or separate mutations. But anyways, so just another way to look at the distribution of malaria in the old world. In places that are wet and low elevation, you have the right mosquitoes to spread malaria around. And so this shows you that in some of those areas in Western Africa, more than 1 in 64 people have this mutant hemoglobin allele. Very common there. Must be a very wet, mosquito-rich area there. So, viewing phenotypic ratios one more time. 3-to-1 monohybrid cross, complete dominance, 1-to-1 monohybrid test cross, 1-2-to-1 monohybrid cross, incomplete dominance, 2-to-1 lethal alleles, dihybrid cross 9331, dihybrid test cross, and then the crosses from epistasis. And from chapter 5, I want you to do problems 22, 23, and 36. And I'll send out an email as to when they are due. And thanks. We'll talk to you again soon.